presently now have 20, we have 19 participants. So we'll try and get everyone participating in the poll.
Yes, please answer the poll. So yeah, let's see how the distribution will be. As you also have seen, there is a pop-up notice on the screen that this meeting will be recorded. So I hope that is fine with everyone. Okay. Let's close this first poll and see the results.
Okay, we have 11 people who've answered, so maybe just give it a few more seconds.
Okay,
oh,
that's
12.
Fine.
Okay, I'm going to end the poll. Share results.
Okay, so most of you thought that it is about 32%, and that is actually the correct answer. That first option is for agarwood or Aquilaria species, which are also CITES listed important species here in the region. 12% is for rhinos, and 41% was the situation for rosewood a few years ago, so between 2009 and 2030. So we can see that the share of rosewood species of the illegal trade has come down. It doesn't mean that the trade has lessened, but what we noticed is that actually the trade in pangolins has picked up a lot of speed, so that has eaten into the share of rosewoods. And I guess that also relates to why we have all been in lockdown for the past couple of years. Pangolin was one of those suspected carriers of COVID, isn't it? Okay, so thank you for your answers for this. Let's take now the next question.
Next poll, okay.
Okay, next question is about Dalbergia oliveri or Burmese rosewood. And we are asking when was the species assessed as endangered on the IUCN Red List? So please add your or make your choice here, and let's see the results then. So we will give it again about half a minute before we reveal the answers.
Everyone's encouraged to participate. We have 24 people who've joined the meeting now, so even if you're not sure, give it a try.
Okay, we'll have another 10 seconds. Make a guess in case you don't know the answer. Okay. 5, 4, 3, 2, 1, and we can close the poll now. Okay. Okay, that's interesting. Most of you actually chose again the correct answer. So this species was assessed in 1998, and already at that time it was endangered. So we can imagine how the situation has evolved since then in those past 20 years. Okay, and let's have then the final poll, please.
Okay. See. Char, are you doing that?
Hold on. It should be launching by now.
Okay.
All right, it's up. Can you see?
Okay, there it is. Thank you. Yes. So Dalbergia cochintensis is probably the most well-known of these Dalbergia species here in Indochina. But how many species of Dalbergia there actually are in the region's countries? So again, please pick your answer and we will see the results in about 20 seconds. There's a very good comment from Nguyen Trinh Le that wildlife should include both wild plants and animals, although it is very true that people normally only think about wild animals, things like rhinos and ivory and so on. Okay, another 10 seconds, and then we will close this poll. 5, 4, 3, 2, 1. Okay, and we can close the poll now, please.
Okay.
Okay, and let's see the results. Okay, this one was more dispersed. Okay, so there is a tie between 25 and 35 species, and the correct answer is actually at least 34 species. So including all the species from China, Myanmar, Thailand, Cambodia, Vietnam, and Laos. And the most species rich of these countries in terms of Dalbergia is Myanmar, where there are at least 20 known species. Okay, thank you for your participation. I hope you enjoyed this brief exchange. And we will start now with a brief video about some of the project highlights, and then we will go into the meeting. So over to you, Char. Thank you.
Okay, I think we can start the meeting. Hopefully everyone can hear me and see me okay. My name is John Mackay. I'm at the University of Oxford in Britain, and I wanted to welcome everyone to the workshop. The sun is just rising here in Britain at the moment, so that music accompanied the sunrise. It was quite nice to see all of you, and it's a real delight to be able to share the outcomes of our workshop today. So what I'll do for the next 5 to 10 minutes is share my screen. I have a short presentation, and then we will move into our different sessions for the workshop. So share screen. Okay, hopefully that should work. Can you see my presentation in presenters mode?
Yes.
Okay, thank you.
Yes, 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 yes.
Thanks. Okay, so
John,
yes.
the presentation is in notes mode, so we can see your notes.
Okay, okay. Let me do it differently then. Not sure. Hold on a second.
Display settings, John. In display settings at the upper portion.
Yeah, hold on. So if I do it this way, oops, is this better? No?
Can you, yeah.
Okay, so I'll do it this way. For
Yeah,
some
it's
reason I can't.
better.
Yeah,
Okay.
it usually works fine, but let's see if I can.
It's good.
Okay, so I just wanted to welcome everyone, and as you've seen already, this project's involved people from several different countries. So I just wanted to start by highlighting who makes up the project at a kind of a high level. You'll be hearing from each of the individual partners today, so I won't introduce everyone, but I just wanted to put up the slide to say that we've had a coordination team of Rena Delonen, David Bosher, and myself. The project has been running for 3.5 years, and we've had the support of a monitoring and evaluation advisory board made up of 5 members, and I know some of them are present here at the meeting, and some of them will be talking to you later. So why should we worry about conserving Asian rosewoods? Well, we already have seen the statistics this morning. Rosewood is the single most the species or the group of species that's subjected to the most illegal trading across all plants and animals. So rosewoods come ahead of elephants and rhinos and all other groups. So they are under threat. And today in the workshop, we'll be talking about 3 major areas that are related to conserving rosewoods, and we'll look at this from 3 different angles. So the first theme of the workshop is to try and understand the threats to rosewood populations, and also very importantly to their genetic diversity. And their genetic diversity is essential for the survival of the species and also for its sustainable use. And that's what we'll be talking about in our themes 2 and 3. So first of all, how do we conserve? We can set up conservation units. There are some that already exist. We want to consolidate those and most importantly, develop new conservation units so that we can preserve these genetic resources. So we refer to these as forest genetic resources. the genetic diversity that exists in the species. And thirdly, how can we use these forest genetic resources to enhance livelihoods of communities that have these trees and that are willing to work to preserve, but also produce seeds and seedlings from them. So that gives you an idea of why we need to do this, how we're approaching it. And so in the workshop today, we'll have these three themes represented by two talks each. And what I'd like to highlight now is the fact that the discussion will involve two of the members of our monitoring and evaluation committee, Chris Kettle and Kim Sobon. So I wanted to thank them and just let you know that they'll be giving some live commentary on the impacts of the project. And so with that, I just want to again thank you for joining our workshop. I hope you enjoy it. We ask you to mute your microphone unless you're speaking. So if you want to ask a question, you can unmute your microphone. Certainly turn on your camera if you are talking. Be aware that using the camera can decrease the bandwidth, so only use it when it's needed. If you have a question, you can raise the hand function, use the raised hand function, or you can type your question into the chat. And so that's it for me. I'll stop sharing my screen and we can introduce the first group of speakers. Okay, thank you, Char. So for the first session, we have Hannes Gaisberger, who's from Alliance Biodiversity, the Alliance of Biodiversity International in SEAT. He will be talking about threats, mapping of threats and assessment of conservation needs. And then Henry Hung, who's a PhD student at the University of Oxford, who will be talking about genetics and specifically about the use of genomics to understand adaptive diversity. So what part of the genetic diversity is important for adaptation? So quite two fundamental aspects to help us improve our understanding and setting priorities for conservation. So with that, I think I will let Hannes begin his presentation. There'll be 5 minutes for questions at the end of each talk. So you can use the chat to write your questions. Thanks.
Yeah, thank you very much, John. So hello and welcome everybody also from my side. I'm actually really very happy to be together with you again, even though only virtually this time. And sorry? Can you hear me well?
Yes.
Okay, good. And I'm going to present you the results of a range-wide vulnerability mapping to identify the species-specific priority areas for conservation and restoration. And actually, the results of this analysis in a paper is currently under review in a peer-reviewed journal and will hopefully be published very soon as well. So, yeah, sorry, maybe for those who don't know me, as John already said, Hannes Gaisberger from the Alliance of Biodiversity International and SEAT, and I'm specialized in GIS modeling, vulnerability assessment. And yeah, so why or let's say what we know at the moment is that in general, there are multiple anthropogenic threats that drive local extinction of tropical tree species. This is in particular true for these high valued ones like the rosewood species here in our study. And in addition to global warming, the forest habitats in the Greater Mekong are being rapidly destroyed by industrial scale, mostly illegal logging and agricultural expansion. And the resulting loss of genetic diversity limits on one hand the opportunities to restore viable populations, and on the other hand also it reduces the species' ability to adapt to a changing environment, both very, very important functions. So what we did is we applied a spatially explicit multi-threat framework that we developed and refined and assessed the vulnerability of the 3 rosewood species to 5 key threats across their native distribution range in 6 countries of the Greater Mekong. These threats were overexploitation, fire, overgrazing, habitat conversion, and climate change. And this spatially explicit multi-threat framework, we initially developed it in 2017. Here are 2 publications if you're interested in further details. And I would like to mention at this point as well that just last week, we also got the vulnerability mapping exercise or analysis accepted or published in conservation biology for the Apfogis project, who was looking at the entire range of let's say, of a broader range of species in South and Southeast Asia. That's also very good news. So what we did here is we used MAX and habitat suitability modeling to estimate the distribution ranges of the species and also the exposure to climate change. Then we employed freely available spatial datasets to estimate the exposure also to the other 4 current threats. And finally, we developed or we calculated a sensitivity score using functional trait information and created the so-called vulnerability maps. Here in this table below, you see all the data sources to create the exposure layers. So I'm not going into any detail. If you're interested in some, there is this question and answer session directly after my presentation where we can clarify or ask, where we can ask any curiosity or any question. So based then on the threat levels, here the current threats and the climate change threat, we identified species-specific priority areas for conservation and restoration for these 3 species. And on the top left corner, you see the priority map for Dalbergia cultrata, where we identify areas prioritized for in situ conservation, restoration activities, and ex situ conservation. Compared to all the previous studies, what is really new here is we subdivided these areas by ecoregions as surrogate for genetic diversity, so as proxy for that to capture also the adaptive variation within the species. And an ecoregion was defined as high priority for conservation of this adaptive variation when less than 10% of the potential distribution area was stable and inside of protected areas. So this was our conservation target in this study. You see here on the left, again, the distribution or the model distribution area of Dalbergia cultrata and the ecoregion presence. So coming to the results or the main results at the species level. So what we found is that all 3 species have significant threat levels in more than 80% of their natural ranges. So this is not very surprising, I guess, for many of us. Protected areas cover only between 11 and 20% of the species' predicted ranges. This might be already more surprising, I think, at least for myself. We found that overexploitation was the single most important threat, followed by fire and habitat conversion. And interestingly, using the new climate change data from the 6th assessment report, because this is the newest and most updated climate change data sets available, we found that Dalbergia cultrata is severely threatened by climate change, whereas for Oliveri and Cochincinensis, it is looks to be less of a threat. What is in a way very much in line also with the findings of Henry, who will give his own presentation directly after this one, who found in one of his studies on Cochinchiniensis and Oliveri that they respond actually different to drought and to heat stress. And here we see the proportion of suitable habitat prioritized for these conservation and restoration activities. The protected areas, the in situ, ex situ, and restoration. What is, most interesting part is here, a very large area is prioritized for restoration, and a bit different the situation for ex situ, for cultrata. Now coming to the intraspecific level, about half of the ecoregions were classified as high priority for improving the conservation of adaptive variation, so quite a lot. And none of the species is sufficiently protected within individual ecoregions in individual countries. I've shown here one example of these high priority ecoregions, the northern Indochina subtropical forests, because it's one of the 3 ecoregions where all the 3 species are supposed to occur together. And we identified 3 possible follow-up actions to improve the status of these areas. And the first one was to designate additional protected areas or conservation units in areas that are prioritized for in situ conservation. If this is not enough, or if there's not enough area, or if this is not possible, then the next step would be to restore the species populations in areas prioritized for restoration. This is normally either degraded or converted land that remains suitable for the species under climate change. This is very important. And last, there is also the possibility of conserving the species ex situ through seed collection and field gene banks. And in the case of the northern Indochina subtropical forests, we see that these possibilities for extending the protected areas to further areas priority for in situ conservation is relatively large. For cultrata again, about 19% of the species potential distribution is falling into these areas. These are the green or maybe the light green areas here in the map, where we see the number of species prioritized. There is a huge possibility for all the 3 species for restoration in this ecoregion. And interestingly, for Dalbergia oliveri, also quite a big area priority for ex situ conservation, although it's not the most threatened species by climate change, as we saw at the species level. So it's giving a different picture here at the smaller scale. So finally, the main recommendations coming out from all that is basically, when we look into in situ conservation, it's very important to have additional protected areas. And it's most affected in areas where the habitat is very suitable in these, as I described before, in these priority areas for in situ conservation. Because of the high genetic differentiation occurring in the species in general, this was found, for example, and not only of course, but by Ida Hartwig. It's also very important that the conservation needs to be expanded also in the underrepresented areas to avoid really the loss of unique adaptations. The restoration activities outside of the current protected area network is necessary for recovering and maintaining the adaptive variation within the species across large part of the ranges. So we saw a huge part of the area is actually under high threat and needs some action. Ex situ conservation, it's important here to focus on areas where the climate threat levels are high, but the current threat levels are low, because in those areas it's very likely to find viable remnant populations. And in general, we can say that spatial analysis is really an ideal complement to field studies in targeting areas with high potential for conservation and restoration, and finding most synergies between species and countries within ecoregions. Because at the end, better targeting saves resources, not only financially, but also time, because this is really highly important in the race against extinction of the rosewood species or also other severely threatened species. And if you're interested in having a look at the more detailed vulnerability maps or priority action maps, please have a look at our website, www.treediversity.org, where you find those results of the maps in Southeast Asia for the Apfelgis project. And as soon as also this article will be accepted, also the detailed maps of the 3 rosewood species in Indochina. So thank you very much for your attention. And yeah, I'd say the floor is open for any questions.
Thank you, Heinis. That was really interesting. Thanks for sticking to the time. I don't see any questions in the chat right now. I don't think anyone's raised their hand. I'll start off with a question. So you nicely highlighted kind of the hierarchy of in situ, ex situ, and restoration needs. If we look at Cochichinensis and oliveri, you mentioned that restoration there was a huge need for restoration. What do we need for restoration to be successful? I'm just wondering about the availability of genetic resources and seeds. What should we be thinking of doing to preserve what is still there so that we can set up that restoration?
Is this a question for me or better for the colleagues who have a... I mean, I can give, of course, yeah. Maybe let's go back a bit. Maybe I'm sharing my screen again, sorry. To go back maybe to slides. I think now you were referring to these areas for restoration, these high need, or let's say areas that are highly threatened. I mean, with this type of study, on one hand, we saw that it's a spatially explicit one. So you see, we have a good idea of where exactly those type of interventions need to take place. Here, for example, I mean, that's not a very zoomed in map, but all these type of analysis were done at the resolution of about 4.5 kilometer pixels. So it's relatively accurate. So we can zoom in much further and see where actually those areas are likely to be. So to check, of course, with field studies, because this is what we get from a, let's say, spatial analysis point of view. And the next thing I think is important here, that we have used these ecoregions as proxies for a genetic diversity. So when we see that there is a restoration need in one of these ecoregions, so we better try to find populations or seed sources in the same ecoregion that are most likely to be adapted to the situation there. And these restoration activity areas also take into consideration climate change, or at least what is supposed to, I mean, what we predict about climate change. You know, we can only use the most up-to-date climate data. And this is actually area supposed to remain suitable for the species also in a longer term. In this study, I had a look at climate change until 2050 only, because mainly if it goes further into the future, basically also the reliability of the data is decreasing. So for the moment, we had a look at 2050, but we used actually the most up-to-date climate predictions. So this means basically I had to redo the analysis, because when we started with the project in 2018, the first modeling was done with a previous climate data set. And in the meantime, a new area of climate data came up. So you don't need to wonder, there is a little bit of difference now in the modeling results, because I used again now a new set of climate data for these.
Okay, thanks. There's 2 questions that came up in the chat. Maybe we'll just take a minute to look at the first one. So Hugo wrote, based on the mapping results, can we come up with some specific areas, regions, and specific measures of protection? So you've already talked about the regions, but what about the measures for protection for those areas, taking into account that people already are living there and are in need of livelihood? So that's a challenging question, but do you have any thoughts?
Well, I mean, there's lots of studies been going on what are actually effective measures for protection. And we know, and what we saw also in the field visit, this species is a highly valuable one. So it needs a very active protection by people. So we just can't let it somewhere remotely alone under the protection status. So people need to be definitely part of that. People being aware that the resources are extremely valuable and need to be protected. So in other studies, and we mentioned that also in our articles, of community forests in close connection actually with the youth. And I guess this will come up later in the meeting today. So I would basically say, yes, protection, of course. And we know now more or less where it would be most effectively. But yeah, a second very important part is, and this goes much, let's say, above what spatial analysis can do. So we need to have people part of that who are actually aware of the importance of the species and that help really to conserve them, to conserve these valuable resources, be close to the sources, and yeah, help us in protecting them. That would be my answer, but probably Rena and other people have more detailed thoughts on that as well. But definitely we saw that we can't leave these tree species just in an area protected somewhere, what is maybe much easier in Europe, where you just, let's say, leave the nature where it is, but because people will find these high valuable resources till this illegal logging is such a dominant threat.
Okay.
Very specific or particular situation, I would say.
Okay, thanks. Thank you, Hannes. We'll move on to our next speaker now. So thank you very much for your presentation. We'll capture the questions in the chat, so we might have opportunities to answer them later on. So the next speaker is Henry Hung, and he'll be talking to us about his PhD work on genomics of Dalbergia cochichinensis and Dalbergia oliveri. In light of Hannes's presentation, it would have been interesting to have Coltrata also, but maybe for a future project. So Henry, when you're ready, you can start sharing your screen.
Sure, thanks. I think Charlene needs to unshare before I can share my screen.
Yeah.
Okay, thank you. Yeah. Is it showing up correctly?
Yes.
Perfect. So greetings, everyone. I'm Henry Hung from the University of Oxford, and it's my pleasure today to talk to you about genomic prediction under climate change, specifically using their tools for matching germplasm to our restoration goals on the two species only, cochichinensis and Dalbergia oliveri. So I'd like to start with briefing what genetic diversity is, in case this is a new term to you. So genetic diversity is really the fundamental unit of biological variation. And on top of it, it's species diversity, our biodiversity we are looking every day, and the ecosystem diversity. So we have forests, we have shrublands, we have river systems, for example. And genetic diversity is also the basic unit where adaptation works on, so adaptation with the environment. And why are they important? I will give you a quick example. For example, in a forest, we have trees with the same genetic makeup that also means the same tolerance to a particular temperature, for example. So if this temperature suddenly changes, let's say in a decade, it heats up. And because all these trees have the same tolerance, all of them will be wiped out, and these species will go completely extinct just because of the climate change. So that's why genetic diversity is important, that confers a bit of variation in the adaptation so that they can endure the future changing climate or just changing environment. Now we have said that genetic diversity is important. Where are usually the genetic risks in the conservation or restoration project? like ours. So it all starts with the natural populations where we get seeds for propagation, and then we have these seedling young trees, which are more commonly referred to germplasm when we distribute them for different forestry end uses. So one of the examples of an end use is to bring the germplasm back to the natural populations for restoration in situ, in hope to reduce the overharvest pressure. And on the other hand, it could be for assisted colonization in a new area for the purpose of forest landscape restoration, for example. And also in our project, we are hoping to engage the indigenous people or the people in the community through income for their resilient livelihoods through income generation, so that we can get people involved along these germplasm supply chain here. But where are the genetic risks? Well, so the first genetic risk could be an artificial bottleneck when we undersample the adaptive variation in the natural population. So that could be the first bottleneck. So along the chain, the genetic diversity is compromised. Second, when we bring the germplasm back to the natural populations, the germplasm may be maladaptive, compromising the survival of these germplasms and thus the effectiveness of the restoration program. And of course, due to climate change, these maladaptation may happen in a later time frame, and especially when we bring these trees to new areas, climate change may alter how these trees will survive in long term. So these are three main genetic risks along the germplasm supply. So I guess the big question will be, how do we ensure a sustainable supply of genetically and adaptively diverse germplasm for forest restoration? And these projects really nicely captures the two sides of the coin, two sides of it. So we have social economic studies in this project to understand the sustainable supply part, while my work is more focused, and also what you heard from Hannes, focus more on how to capture the diversity in the germplasm across the region. And particularly in my work, because of the long generation time of trees, we can't do experiments to test the adaptiveness in this germplasm. And that's where we think using genomic technologies to screen the individuals will give us very rapid decision-making evidence. So in particular, the first thing we want to address is the genetic bottlenecks, right? And special shout out to Ida Hartwig. I think Ida is actually among us now. I think she is here. So special thanks to her. So her work in 2017 laid a very important foundation on understanding the genetic diversity of these two species across the range. And she delineated 5 main conservation units, as you can see in the figures here, that each color is the dominant type, genetic type for that particular area of population. So you can see some landscape differentiation between populations with different genetic methods. My work kind of tried to probe deeper using 200,000 genetic markers and only focused on the adaptive side, the adaptive component of these genomic variation across the species range. And I not only assess the genetic variation with the samples we got, but I also predict that adaptive variation across the entire range, the entire species range is predicted in the Hannes study. And as you can see, it's color-coded here that how the variation is distributed on the region. The left-hand side is the congensis, and right-hand side, the olivary, and those black dots are where the genetic samples we got are from and when we do the genomic screening. What's also interesting to look at is actually in this exercise, we also found that congensis is more temperature-driven. The genomic variation is more temperature-driven, while for olivary, the variation is more precipitation-driven. And this kind of correlates with a previous study we conducted in 2020, published in Ecology and Evolution, that we conducted a greenhouse experiment to test the drought effect of drought on seedlings. And we found that for there's a contrasting difference between the two, where in terms of water relation, cochinensis shows more variable changing water relation, but in contrast have a higher sugar content from photosynthesis. And we think that this is related to the ecological niche, where cochinensis is drought tolerance. It's a pioneering species, so it maximizes growth at its young age, while olivary is more at the climate species and try to avoid drought. So that's why cochinensis is in our genomic exercise is more temperature driven, while olivary is more precipitation driven. So to address the second issue, the adaptation. So one important parameter we are thinking here is local adaptation. So we assess the strength of local adaptation in these two species, and we found particularly strong local adaptation, or shall we say gene development association, in coastal areas in particular, color-coded by those blue regions there. Local adaptation means that the germplasm, the genetic materials there are strongly associated with the environment there. So it's very likely if you bring other external materials there or bring them out, it will compromise the association, the adaptation. So these areas are particularly recommended for in situ conservation, and of course at a higher priority, because if this association is lost, it's hard to recover. And finally, third, it's the climate change we have to address. Trees have particularly long generation time, which is thought to be more easily outpaced by climate change than other biological organisms. So populations may be adapted to the current climate, but they may not keep track of changing climate. And that's where we introduce a metric known as genomic vulnerability, which measures how likely the population will become genetically maladapted in the future, using the same climate scenarios as Hannes already introduced to you. So, for example, we know that we want to restore some in situ populations, also known as the seed sources. And if we know that geographical locations, we'll be able to predict how the genetically maladaptation may happen in the future, and that's useful for priority setting. And of course, highly vulnerable populations should be conserved with higher priority, because that means that these are where the species that most likely to be maladapted the soonest or the severest. So in cochinensis in particular, well, this is one of the scenarios, one of the possible scenarios using the SSP 370. And as you can see, the changing time, the increasing time. Well, as expected, as the time progresses, the genomic vulnerability increases as the environmental values differ more from the current state. But what you can see here is cochinensis doesn't really see a distinct pattern of genomic vulnerability. These may be related to what Hannes just mentioned in his presentation about that cochinensis is less affected by climate change. However, on the contrary, olivary has more distinct genomic vulnerability pattern that some areas in particular have a higher likelihood of maladaptation under climate change. So these may be taken account into when we're setting our conservation priority exercises. But now, as we know that in these areas, we want to do ex situ conservation, we want to move the germplasm materials out because they are more likely to suffer in the future climate. So that actually changes to another side of the coin. Another side of thinking is to think about genomic match, genomic similarity, where we can assess the match between the current seed sources, those populations that have higher genomic vulnerability, to adapt
Henry,
to the future.
Sorry to interrupt. I just wanted to let you know you have 1 minute left.
Okay, yep, it's coming there. Yep, thank you. To a future restoration site. So let's say we have in mind some ex situ populations and uncolonized sites. We want to move our seeds to there. So again, we can use geographical information to predict where's the best to get the sources, the seed sources from. And this is known as assisted migration. We are trying to help the species to pre-adapt to the changing climate. So actually, the one very direct output, and actually wrapping up this presentation, is an online application for genomic prediction that's under development now, supported by Oxford server and developed in completely our language. So we call it Cinder with the R as the language we're using to write this, that I'm using to write this online app. So I can't really show you a real demo now because it's still under development, but hopefully it will be released soon. But let's say, for example, we know a particularly forest restoration site we have in mind to restore our forest, for example, 106 north and 14 east, a particular longitude and latitude. And if we choose a particular scenario, climate scenario and time period, we'll be able to predict how the genomic match looks like to the current germplasm, current distribution. So the right asterisk case, our designated restoration site, while we know the similarity across the range for now, for current seed sources across the entire range. So we hopefully will release this soon, so please stay tuned. So really the conclusion is, to address genetic bottlenecks, make sure the adaptive variation of the population is well represented in our germplasm collection exercise. On our adaptation, germplasm with strong local adaptation are recommended to conserve in situ. And finally, climate change, populations with high genomic vulnerability should be conserved ex situ, and assisted migration by matching current germplasm at future restoration site may help the species pre-adapt to the changing environment. And this is the three main publications we've done so far, and we hopefully will release 2 more in the coming year. But for any questions, please email me. Thank you very much.
Okay, thank you, Henry. We have about 2 minutes for any brief questions. So maybe just reflecting on your finding that the vulnerability for Cochinensis is spread across the species range quite uniformly. That seems to coincide with the findings from Haynes's work as well. So what are the implications for conservation in that species?
In particular, Cochinensis,
Yeah.
right?
Yes.
So I think that may be also because of the from the study from Ida Hartwig from 2017, we see a more stronger spatial structure in the genetic diversity, and also actually in my study also in adaptive diversity. So it seems spatial factor is very dominant in Cochinensis in particular. I haven't looked really deeply enough to understand, to have a plausible explanation for this, but could be due to their dispersal range, for example, or clonality and clonal distance. But well, seeing the spatial structure is the most important factor. I will argue that genetic bottleneck may be the most important risk, or most likely risk for Cochinensis, and that's where we should think about how these spatial factors work, the scale it works, and then the setting up conservation units or setting up germplasm collection exercises based on the scale of these spatial factors are working on.
Okay, thank you. I think we'll move on now for the sake of keeping on schedule. So thank you, Henry. Thank you, Haynes, for your presentations. I just wanted to comment. A few people have been asking about sharing presentations and maps. So Haynes has already responded to that. We'll talk to all of the speakers and see what can be made available in terms of presentations after the workshop. So at this stage, I think David Bosher will be taking over for the second part of the workshop. Thanks.
So good afternoon, good evening, good morning, wherever you are, different parts of the world, different time zones. The next series of speakers, next 2 talks, are going to be covering aspects to do with the second theme of the project. So the project was roughly divided into 3 themes. We've seen, first of all, about threat mapping and issues to do with adaptation. And so the second theme that we're going to cover is talking about filling conservation gaps, the gaps that have been identified through new conservation units. And we're going to have two talks, one from the work in Laos, and the second from the work in Vietnam. So first of all, I'd like to introduce Chaloon Bunifiphon, who's from the National Agriculture and Forestry Research in Laos PDR. So
Hello,
welcome, Chaloon, and over to you.
Okay, thank you very much, Dr. David. Hello, good morning, good afternoon, everyone. I'm Chaloon Bunifiphon, working at Forest Research Center, NAFRI, under MAF in Laos PDR, and I also a part of assistant for the project coordinator. May I share with you my screen? What I'm going to share with you or introduce to you today is about the development of Rosewood Conservation Unit in Laos PDR, especially in natural forest. First of all, and sorry, thank you very much to give me an opportunity to share information about Laos and also as well as the Rosewood of Laos to this event. I'm going to talk about 5 contents here, and the first one is about the status of Dalbergia tree species in Asia. As you may know that there are a lot of species under genus of Dalbergia species across the world, especially in Asia, Africa, and Central and South America. And for the Asia, there are 119 species occurs in Asia, and you can see that in Laos PDR or in the region here, we have the very intensive occurrence of these species. Okay, here is based on the result of development of the global potential distribution of Dalbergia genesis from after this project. We can see that there are the potential distribution of the Dalbergia genesis across the protected area and potential distribution, and also potential distribution in converted area. For this species, we can see the potential distribution in convert area in Laos, not too many, and also the Dalbergia oliveri also, but the potential distribution quite more in Laos, from the northwest of Laos to the southwest, and also the Dalbergia crunchata. And for the second point, second topic is the status of the conservation units in Laos PDR. About 20 years ago, there was a 3Cs project under Danida project. Laos PDR has established 102 conservation units of 3 species in Laos, and there are 2 or 3 sites where of the cogenesis has been established, but unfortunately, due to the development of Laos PDR and also other trade, that area has been converted to others' land. And currently, in 2018 or 18, the recent assessment of the 3 seed sorts has been found that there are 76 tree resources are remaining. The cost and the treat, I mean, the cost of the treatment is mainly is from development program and livelihood. Illegal logging, it means that like overexport, and also low incentives and motivation of seeds demand, and significantly is the high market demand, high demand market of rosewood. So those are the cause of that force that or it negative impact to the decreasing of the Dalbergia population in Laos. Under the Dalbergia project, Dalbergia Darwin project, we have established 2 ex situ and 2 in situ conservation sites in 2 districts, in middle to south. I mean, in the middle of Laos, in Savanakhet province, and 1 germ trust collection of 3 Dalbergia species will be established at Forest Research Center, about 2.5 hectares. And within the project, we have assessed the status of the population of Dalbergia tree species in October 2019, and we have found that this is just the we have assets in focus. We just focus on the most occurrence population of the Dalbergia tree species. We have found that the population of Dalbergia congensis is really low, and also the range of the diameter size very small. And for second communities, we also found that Dalbergia congensis and Dalbergia punjata is very small number of population of their population, and also the range of the diameter is also very small. We found just only the sapling, not the major tree. Major tree is very small. And also the third community is very small and small number of the population, and also the range of the diameter is also very small. Mainly with the highest diameter at base height, we found that 7 to 10 centimeters. That's only three in the area about 10 or 15 hectares. And according to the result of the assessment, what we have action to recover or to restore. We first, as I said, we have implemented baseline study, and second, we have inventory, assess the population, and second, we mainly support communities on capacity building, especially training, and finally we make we establish the in situ and ex situ conservation and germ price correction. What we have done is we have trained communities, people in communities, and also people in forestry and agriculture sector, and also private sector in local area to know on the how to collect seed and how to germinate and how to manage the nursery and produce seedling. The seedling that we have produced, we distribute to those involve family in our target, in our project communities. And also we introduce into the ex situ and in situ conservation.
Chaloon, you have 1 minute left.
Okay, thank you. And what we what is here we have two, as I said, two in situ and ex situ. We have quite large area of in situ that we introduce 6,300 for ex situ, in situ, and ex situ about 5,200. And right now we are preparing we preparing the site, and also try to keep this the good quality and the good healthy of the seedling of Dalbergia, three species of Dalbergia to plant in next season. And this is the way forward that we have planned, working with our project team as well as from Dr. David, Dr. John, and Dr. Rina, and our team members to brainstorming and put our target, what we are going to do in next in future in order to restore or to protect our Dalbergia tree species. And yes, and that's all for my question. Sorry, that's all for my presentation. And sorry that I cannot explain briefly about the table. Hopefully you can see my presentation. Thank you.
Thank you very much, Chaloon.
Yes.
Do we have any questions to Chaloon? Oh, I see a hand. Oh, no, it's applause from Hannes. I thought it was a hand up for a question, but it's applause from Hannes. So thank you. Does anybody have a question? I suppose my question, Chaloon, would be of those actions you identify for the future, what is the highest priority to ensure or to follow up on the conservation of the species in Laos?
Yes, according to our discussion and brainstorming, this is not only from my idea, and also not only from Forest Research Center, but also from others organization, especially other department of other department under Ministry of Agriculture and Forestry. The main priority is we need to conserve the specific habitats for the species for in situ, to make the in situ and ex situ conservation, and also to make a good source of material for germination or seedling production, something like that. Yeah.
Okay, thank you, Chaloon. There's a couple more questions come on the chat. One from Ida Hartvig says, Chaloon, thanks for the presentation. It's worrying that only small trees were found in Savannah Ket. And how many sites were you able to collect seeds from?
Actually, it's really hard to find nature tree in natural forest, but we can find in other lands. For example, other lands mean that outside forest, tree forest category, it means that in farmland of in private land, we can see in private land quite many, but of course the distance between tree to tree is quite far. It's about at least 100 or 50 meters. Yes, that's we can collect seed from from their natural habitat.
Okay, thank you. And one final question from Hugo. How do you get these seedlings to grow up vertically rather than bend down and grow horizontally?
The seedling that we got is we collect from, from not in the same area, but nearby the same area. I mean nearby where we introduce where we planting the seedling into the say into the ex situ and in situ. Yeah.
Sorry, everybody. Well, thanks very much, Jaloon, for a very interesting presentation. We're going to move on now to our second presentation, which is Dr. Tran Thi Hoa from the Vietnamese Academy of Agricultural Sciences. And Hoa is going to talk about conservation units of rosewoods for local sustainable development in Vietnam. Thanks, Hoa. Over to you.
Yeah, I'm trying to share my screen. Can you see my slide now?
Yes, we can see. Yeah, that looks good.
So good morning, good afternoon, everyone. So today I'm going to talk on conservation unit of Dalbergia species for local sustainable development in Vietnam. So the aim of my talk today is inspire you to recognize important forest genetic resource, especially for rosewood Dalbergia species in Asia and in Vietnam. So think about genetic diversity in the context of management and conservation, and also use our knowledge to develop conservation and management strategy for Dalbergia species for local livelihood. So the content of my talk today, I'm going to talk about distribution Dalbergia species in Vietnam, so conservation status of Dalbergia, and also I want to set out the certification criteria in Vietnam or guideline for establishing conservation unit, and finally for establishment and maintain conservation unit for local sustainable development. So you have already heard about Jaloon, about distribution of Dalbergia in South Asia, but in my talk, I only focus on distribution of Dalbergia in Vietnam. So most of Dalbergia species in Vietnam is distributed in the south of Vietnam, except Dalbergia we consider. So the other species is from south onward. The Dalbergia species distributed both in nature park, nature reserve, and also in farm in a few provinces in Vietnam. But based on our, only remain in scattered individual, or by using farmer interview and inventory, no more individual at present in farm. So you can see in the picture here, olivery is cut down for illegal harvesting, even for root. So we can see very heavy and dangerous for Dalbergia in Vietnam. So where the population remains, so mainly it distributed in Joc Don, in Chu Giang Si and Chum Roi Nature Park. But in Joc Don, mostly based on our Darwin project, we based on our survey, almost the tree from forest have been moved to the arboretum for ecotourism. In Chu Giang Si Nature Park, is distributed in scattered individual of jungles. We can see here about 7 kilometers between trees. You can see here our colleague. So we can see very heavily situation for the remaining of individual. just very young leaf or seedling in the very heavy stone. So the only tree population remain in Chum Ray National Park. That's why we choose Chum Ray National Park as case study for our Darwin project in Vietnam for conservation of Dalbergia species for local livelihood. We also set our certification criteria or guideline for establishing conservation unit. So it's agreed by our project team, our local government, and also our colleague in both institution and also in a local team for Darwin project. So based on our criteria agree, so we established 3 sites for conservation unit in Chum Ray National Park, both for Dalbergia oliveri. Let me take my power. For oliveri, for Cochin sinensis, and also for in situ and ex situ conservation unit in Chum Ray National Park. You can see in the picture here. So in situ site for Dalbergia Cochin sinensis and Dalbergia oliveri, we have selected the 30 hectare and approximately of number 3 we can count it 503 with 23 of 20 years old. And the second site for ex-situ site for Dalbergia Cochin sinensis, we have selected 3 hectare for both species. For Cochin sinensis, we choose 153 and another species, Dalbergia oliveri, for 63. So we can see in the map here, we can see it here's oliveri. In local name is we call Cầm Lai, and it is area for Cochin sinensis. Vernacular name is Chak. So the activity in our conservation unit, like Laos and Cambodia and another conservation unit, we have field survey for completing the conservation unit protection. We also, by using funding and implementing of Darwin project, it's first time we collect the seed of Dalbergia oliveri and the seed of Cochin sinensis that we have never done before. So it is a good opportunity for us and also for farmer to start with seed collection and marketing the seed of Dalbergia in collaboration with Forest Genetic Center and private nursery. We also have field survey for find out the disease and apply the disease control measure, and then try to improve the seed qualification, especially for Cochin sinensis, but not heavily for oliveri. But we need to have disease control measure applied for Dalbergia Cochin sinensis. And the most important thing by implementing the Darwin project is that we establish the new nursery at Chum Ray National Park to strengthen the seed source. It's also for conservation. You can see here's a picture of the conservation unit in that town. So with Dalbergia oliveri, and then collaboration with our staff, and also Chum Ray National Park, and also farmer, we collect seed from 2 species by using the traditional methods. We climb up with a tree, and also we use the technique from Dalida Forest Center with the technique how to climb and how to collect the seed successfully. So we have collected 1,000 Dalbergia oliveri in year 3 and year 4, and then and also between 300 and 400 seed of Dalbergia coccinellus in year 3, and we continue to continuing to research and collect the seed in year 3 and onward, based on our conservation plan for 5 years by using funding from different source of government and another currently project. We also have set up training for seed collection in farmer and staff. So we have 7 training in 3 years, year 2, 3, and 4, and continuing, and also the same for marketing the seed in Dalbergia species. You can see here it is in Bargok, in situ conservation, and also our Chumray staff. So we have a meeting and for training and transferring our knowledge on seed collection, and also scientific trainer also come to work with us. And we have
one minute, please.
Yeah, thank you. I have almost done. So we have new nursery established in Chumray National Park. You can see here it is collaboration with our staff, with veterinary and also farmer for our new nursery. So it's end of my talk. You can see here we have successfully to collect the seed from Dalbergia oliveri, and also for Dalbergia coccinellus. So thank you to all for interest in my talk, and thank you for the Darwin funding for our project. And we have opportunity to start with seed collection of valuable Dalbergia species in Vietnam. Thank you.
Thank you very much,
Yeah.
Hoa, for your interesting talk. Do we have any questions to Hoa about her talk specifically, or any more comments, maybe general comments about the area of conservation units within the region, or conservation units as a means of protecting genetic resources of species? Maybe I'll make one comment, which I found interesting in your work, Hoa, was that, as I understand it, this was the first time the permission had been given to grant to make collections from the species within the national park. So that required a change in regulations to allow that to happen.
Yeah, it's first time to allow the farmer to collect the seeds from national park. It is a very good collaboration between local staff, local government, and also our project team. People not allowed to cut the tree down, but you can see here we use the technique learned from Danida Forest Seed Center, and also another traditional technique to collect the seeds from very high tree.
Mm.
Yeah.
So I think this is a very interesting issue, that in terms of when we think of in situ conservation, that there are times when it's just viewed as something that is locked away, and therefore there is a disconnection between the conservation of that resource and the contribution that resource can make to livelihoods. And I think that was an interesting thing that happened in the project, with you being able to change the regulations to make that collection happen.
Yeah. Yeah.
Okay, I don't see any questions. So I think the next thing on the schedule is a quick comfort break. I think there's going to be some polls coming up during that you can participate in, slideshow, and then we should be coming back together again in 5 minutes, if that's okay for people. So we'll see everybody in 5 minutes' time.
Okay, hello everyone. Welcome back from the break. Can you hear me all right?
Yes, very well, Rina.
Thank you, Hannes. Okay, so in this third and final thematic session of today, we will be talking about seed and seedling production strategies for income generation at local communities. Rosewoods are so valuable, and that also means that the seeds and seedlings are very highly valued and much sought after products. And so we will now be hearing about innovations and activities that have taken place during this project in Cambodia and in Laos. And again, there will be 5 minutes for questions after each presentation. First up, we have Dr. Sotea, who is the Deputy Director at the Institute of Forest and Wildlife Research and Development in Cambodia. Sotea, please, the floor is yours.
Yeah, thank you, Rina. Can you see my screen?
Yes. Can you put it on presentation view?
Yeah, I try to put it. Is it okay now? Yes.
That's perfect.
Good morning and good afternoon to everyone. I would like to share experience on a farmer supplier in Cambodia. This is the content of my presentation. In Cambodia, we have placed more than 500,000 hectares of natural forest under the management of local community in the form of community forestry. And this is with the aim to maximize the contribution of sustainable forest management to poverty alleviation, enhance livelihood and equitable economic growth. For this project, we work in 4 provinces. And the first province, we work with the farmer supplier, and we have a provenance trial of Dalbergia Koshang sinensis in Siem Reap province and in situ conservation of Dalbergia Koshang sinensis in Kompong Thom province and in situ conservation of Dalbergia oliveri in Kompong Chnang province. But in this presentation, I will talk mainly about the site number 1, which we are working with the farmer seed supplier. This is the farmer seed supplier, Mr. Sok Em, who start this business as a seed supplier about almost 10 years ago. He supplied seed of Dalbergia Koshang sinensis, Dalbergia oliveri, and Pterocarpus macrocarpus, due to the ease of access to seed sources in the nearby community forest and high demand of the species, I mean that the seed and seedling of the 3 species. Mr. Sok Em received training and technical support from the Forestry Administration on seed collection, seedling production, and marketing. And since late 2010, his family main source of income come from seedling sale, not from the rice field. Mr. Sok Em had to purchase seed tree before he could collect the seed. He had to pay to the seed tree owner, whether on the private land or in the community forest. He pay normally around $50 per seed tree to the owner. This table shows the amount of seed and seedling he produced in 2020, and you can see that large amount of seedling of Dalbergia sinensis produced last year. The price of the seedling here seems to be very low because he mainly produced small seedling aged between 2 weeks and 2 months old to the middleman for reporting. Mr. Sokam has some challenges, but the two main ones are insufficient seed source and irregular seed production. And the second one is his nursery was too small and was in dilapidated condition. So that why the Darwin funded project come in to assist him to overcome some of the challenges by providing training to him and to other community forestry member on seed and seedling production and marketing. We also assist him in construction of a new nursery, which increase the capacity to produce more than 100,000 seedlings per year. We also assist him to establish 2 grafted seed sources of Dalbergia sinensis and Dalbergia oliveri on his own farmland. The farmer will graft some more Dalbergia oliveri tree for planting in next year, because so far he had only 9 mother tree. We plan to increase to 15 or 20 mother tree. This is the old nursery in 2019 before the project intervention, and this is the new nursery after the project intervention. The photo showing the training activity on grafting of Dalbergia sinensis and Dalbergia oliveri. And the grafted seed source of Dalbergia sinensis in the second year. Photo on the right hand side is the close view of the grafted seed source. And this is the grafted seed source of Dalbergia oliveri, which was planted this year. The right hand side is the tree under the plantation condition. Based on his experience, we developed guideline for propagation of Dalbergia sinensis and Dalbergia oliveri by grafting in local language and also in English. Lesson learned: advertisement increased sale. And the second lesson learned is seed and seedling business does not work well with the community forestry. As I mentioned in the previous slide, we also work with the community forestry in Kampong Thom province. Because in the community forestry, only elderly people involved in seed collection and seedling production. And it is hard to promote planting of Dalbergia sinensis among smallholder farmers because they have limited land and long-term investment. For the conclusion, I think the selling tree seed and seedling can be a profitable business for local farmer. the establishment of the farmer seed sources will reduce the production cost. The FA has created an efficient way to seed supply while creating sustainable source of income for the farmer. And technical assistance and monitoring from FA is needed to ensure good seed and seedling are sold to the end user. Thank you very much.
Okay, thank you very much, Sotea, for that presentation. Really nice to see how the trees are growing on farmers' land and produce seed in near future. There's a question in the chat from Dr. Chansamone. He's asking what was the purpose of grafting for these two species. So can you maybe explain a little bit more about why the seed source was established through grafting and what was the benefit of that? Thank you.
The reason for grafting, because when we graft, the tree will produce seed sooner than planting the normal, how to say, the nursery grown seedling. For example, if we plant Dalbergia sinensis, we would wait for 15 years to 20 years to get seed from that tree. If we plant, we graft, so that based on our experience in Siem Reap province, the tree start flowering in the second year, and we assume most of the grafted tree in the plantation will flower in the third or fourth year, so that the farmer can only wait very short time. I mean, a very short, because around 4 years to collect, to start harvesting seed. And this is the reason of grafting, just like the fruit tree. Thank you.
Okay, thank you very much for that clarification. Let's move on to the next presentation in this session. So next up we have Mr. Bansa Thamamong from the Forest Research Center at Nafri, Laos. He's deputy there. So Mr. Bansa, please, the floor is yours. We have actually seen him already in many of the slides. He is the person who is standing at this beautiful seed source at the Forest Research Center in Laos in the photos. Yes, okay, thank you. Over to you, Mr. Bansa. Will you be able to share your screen? Okay, Mr. Bansa.
Yeah, I'm coming. Yeah.
Okay, thank you.
Can you see my presentation?
Not yet. Can you share screen?
Okay.
Mr. Bansa, would you want us to share? Okay, we can now see your screen.
I have
That's
problem with internet network is too slow.
Yeah, it's okay. We can now see your screen. Yeah.
Now, okay.
Right, it's great. Thank you.
Yeah. Welcome to my presentation. My topic is seedling production in Lao PDR, focus on the project activity. This is the content of my presentation. the background of the people in the target of the project. For seedling production, they have the lesson learned from the since 2012. They import the seed of the Persia cocci from the Vietnam to plant in their own land. The time the Persia cocci is play very important role for the income to support the family, because Persia cocci in that area, people sell the bark of the Persia to Vietnam as to the produce for incense. For the lesson from the seedling production of the Dalbergia is the first time for them to have learned from the during the project since 2018 to present the conservation of the Rosewoods of okay, the genetic diversity of the cereal of livelihood in the Mekong has been implemented in Laos, focus on in the Thapang Thong and Nong district, Savannah Khet province. Seedling production activity is one of the activity of the projects that encourages the community to participate in learning and being able to produce seedling to support the tree planting in generated income for the family. The objective of the seedling production in the community to able to plant, to participate, to learn to the techniques of the nursery and able to produce their own seedling as the Dalbergia species. In order to provide the seedlings in for enrichment planting in situ and ex situ conservation area in the target area of the project, as well as the generate income from the selling. This is the target of the project in the Thapang Thong and Nong district in the Savannah Khet province. So for seedling production, the Forest Research Center in collaboration with the provincial forestry staff and Thapang Thong and Nong district as a forestry officer to provide seedling production training to the target villages in 3 communities. The participants joined and with these trainings is about 50 people. The training was divided into 2 parts as studied in the classroom and the field practical. For in the classroom, the trainer explained the techniques of establishing the nursery and at community level. The trainer explained the techniques of the on stage of seedling production and had a discussion between the participants. Later, after community have learned the techniques of producing seedlings, in practice, the trainer led the participants in sowing Dalbergia congengensis, Dalbergia contracta, and Dalbergia oliveri in the nursery. In the field practicum, the community has learned to establish the nursery, soil mixture and potting, soil mixtures covers soil from forest is 60%, husk 20%, and manure 20% mixed together and then potting in the plastic bag. Prepare seed bed. They use the sand to prepare the seed bed, and then cleaning the seed, and then seed selection, pre-treatment of seed by soaking the seeds in water for 16 hours, and then sowing the seeds into seed bed and covering them by sand. Seed germination. After seed sowing for 7 days, the seeds start to germinate young new seedling. And then for seedling transplanting, they prepare the after potting the soil into plastic bag, and then they water the soil is quite before transplanting in the plastic bag, and then they after the seedling for about 16 days, they has to produce new leaves on with 2 cotyledons, and then they can take it out and put in the prepare the plastic bag. And then monitoring and care of seedling, watering, fertilizing, weeding, and control. Preparation of seedling by making the seedling strong and durable by opening the sunshade so that the seedlings are exposed to direct sunlight for about 1 month before plantings in the field. For the results, the participants from the 3 communities in 2 districts be able to learn and understand the techniques of nursery and seedling production. Establish 2 nursery for the target community to produce seedling each year, and to be able to supply seedling for tree planting in the area. Communities can produce seedling for 3 species of Dalbergia, about 15,000 seedlings. In conclusion, strengthening seedling production for the participants in 3 communities in 2 districts in Savanakhet province is a task that needs to be addressed. It is an activity to strengthen the community to learn techniques on nursery seedling production, planting, and to raise the community's awareness of the sustainable forest conservation. In addition, it is an alternative for the community to create their own job and generate income for their families. Challenge. COVID-19 expansion directly affected the project activity, where entire the country was locked down and strictly in traveling. So the team cannot implement activity as planned, and some activities has been postponed. During dry season, the target community experienced drought and water shortage. This affects the growth of the seedling in nurseries. Community seedling marketing is still limited due to the lack of experience in seedling service. Recommendation. Although the community has been trained in nursery techniques and seedling production, the district forestry staff must continue to lead the community to provide technical assistance to the community. Establish a seedling market network is a great way to generate income for the communities.
We lost the sound. I think your microphone was muted a bit too early.
Mr. Bansa, you muted yourself accidentally. Can you repeat the one in your previous slide? You're on mute.
Oh, sorry.
I think that was the thank you slide was on the left, so that was at the very end of the presentation. But so thank you very much for this this presentation. Really nice to hear about all the activities that have taken place in the communities in Lao. There was a question in the chat actually to Dr. Sotea, whether there are any networks for seed collection and marketing among the communities in Cambodia. This question has been answered in the chat, but it's actually a question that I could pose to you, Mr. Bansa. So actually this was part of the activities also in Lao during the project to initiate seed networks and get farmers to collaborate on seed production. So could you just explain briefly about the kind of activities that have taken place about seed networks, or what plans you have for the future on that? Thank you.
Okay.
Yes, Mr. Bansa.
Sorry.
Can you answer the question? Can you tell us a little bit about the seed networks in Laos? Thank you.
Oh, seed network in Laos. We have established the seed source. Like as Chalun mentioned, now we have 67 still remaining for seed, 3 seed source. But for Dalbergia Cochin seed, we just established for last year 3 seed source in 3 provinces in from the outside of the target project is 2 and 1 inside of the project. For the seed network, every year before we collect the seed, we have to the district forestry in the district. They just coordinate with the how to say the owner of the tree seed, and to inform when the seed is flowering and what time is the seed mature enough, and then we go there to collect it. The seed network in Laos just not so still limit for Dalbergia species.
Okay, thank you very much for that. There is a question, very important question in the chat about what happens to the seedlings after planting. So Dr. Hugo was sharing his experience from Thailand, how seedlings are planted, but the post-planting care is often missing in Thailand. Can you give just a very brief comment on this? What is the situation? What is the plan in Laos or Cambodia regarding this? I see Cambodia, Sothea has already answered on that. So this is a question to you, Mr. Bansi. Thank you.
What is the question, please?
So what happens? Yes, what happens to the seedlings after planting? How do you ensure that the seedlings are actually taken care of so that they can survive and grow on those planting sites?
Oh, that mean the seedling in nursery or in the planting in the field?
I understand this question is regarding planting out in the field. Yes, so maybe actually it's a bit beyond what you have done.
The seedling after planting in the field. As our project on seedling distribution in the in situ and some in ex situ conservation. In ex situ conservation, they have fence like in the Thapang Thong district. They produce the fence to protect the seedling, and they have to take care of the DAFO stuff. They take care of after planting in the field, like ex situ conservation. For in situ conservation, it just planted in the area, and the people, the community, they know they can they cannot to go and cut the tree in the area. But how to make sure the seedling is grow? This for Dalbergia species in the in situ conservation area where the Dalbergia grows, is grow well, yeah, under in the forest.
Okay, thank you very much. Yes, so in this case, the seedlings have been planted kind of back into natural forest. Okay, thank you. I see there is Chalun from Laos also has his hand up, but in the interest of time, could I maybe ask that you make your comment in the chat, and we can also come back to this topic after the next session if there is time for further discussion. We also have actually more time for discussion a bit later in the program. But now I would like to hand over to David Bosche for chairing the next session. Thank you. Thanks for the presentations and the interesting questions and comments.
So hello again, everybody. We've heard from 6 different talks covering the 3 main areas of the project, which were quite diverse in terms of looking at the distribution and gaps in the conservation of the species, and then some practical actions in terms of setting up conservation units, and then transferring through to the implications and actions for sustainable livelihoods within rural communities associated with these populations of Dalbergias. So this next session, we have 2 people with relevant experience who we're going to ask for commentaries about what are the implications from what they've heard in these presentations for practice and what perhaps are the way forward for conserving high value threatened species in the Greater Mekong region. First of all, we're going to hear from Kim Sobon, who is the Deputy Director of the Department of Forest Plantation and Private Forest Development within the Forestry Administration in the Ministry of Agriculture, Forestry and Fisheries in Cambodia. So first of all, I'll hand over to Kim Sobon for his comments. Thank you.
Thank you, Dr. David, and thank you, Dr. John, that invites me to have a brief interest with interaction. The impact of the project for the conservation and livelihood in Cambodia and Greater Mekong sub-region. Good afternoon and good morning, everyone. Yes, just a general observation. I think this project is very useful for I think all countries concerned about the Dalbergia species, and recently we have also conducted the assessment on the distribution of the Dalbergia cochinchiniensis and Dalbergia oliveri in the North Cambodia, and the result we found it almost extinct from the natural forest, and although mostly we found the distribution of plantation in in HC2 conservation, and we in overall we found the seedling of in previous time, maybe 10 years ago, most people they don't know what is they call the local name is Grenyum. The people they don't know that species, but now they know that species well because of we try promote the plantation of those species. Either we try distribute, even the government have produced the seedling and produce and distribute to the local community free, and maybe several. I think the last few years, the most people they very interested with this species, and even the COVID-19 impact, but the seedling of production according to our record is increased, particularly in 2020 and 2021. The number of the seedling produced is was increased, but however the seed source, I think the seed source still limited. Most of seed that the most seed from our seed source, but some also we import from the neighboring country like Thailand, Vietnam also. So return back to the technique of the graph technique with the Dalbergia and oliveri that Dr. Sothia have proposed. I think it's very interesting that we can maybe it can be expanded for the enlarge of the like seed source that we plan the graph technique. Maybe we can expand to the like community forestry or our seed source area also. Like because we have our natural seed source area, we have 18 sites, but of those only maybe 4 to 5 seed source that is have the Dalbergia cochinchinensis and Dalbergia oliveri, but I think the seed still limited that those three still pursue and that could capability provide seed. Yeah. Thank you.
Okay, thank you very much, Kim Savon. I'm now going to bring in Chris Kettle, who is the principal scientist and global program leader of the program for tree biodiversity for resilient landscapes, and based with the Alliance of Biodiversity International and SEAT. And Chris, we'd love to hear your sort of perspectives on what some of the results from this project and the achievements mean in the wider perspective.
Sure.
Thanks.
Well, thanks, David, and thanks, John, for the invitation to speak very briefly at the end of what I think is an extraordinary workshop presenting the amazing outcomes of the project. I mean, you know, not least in the context of COVID, the video at the very beginning shows how effective the project's been able to be on the ground in the Mekong region to deliver the project objectives related to enhancing knowledge, to increasing capacity development, and improving livelihood opportunities in the context of Dalbergia in these countries. And if we place this in the context of COP26 and the major commitments that countries are placing on tree planting as part of their mitigation and adaptation strategies to climate change, it really even emphasizes even more the importance of this project in the region. I think the framing of the workshop around the three themes, it was really useful and to adopt that in terms of the impacts of the project really in the priority setting. You know, the work that's been done on knowledge generation there has been hugely valuable in enabling us to understand better not only the relevance of climate change in its role as part of the threat to the conservation of the genetic resources of Dalbergia, but also the interaction between climate change and other land use and over-exploitation, and very clearly identifying that protected areas are clearly inadequate for the conservation of the genetic resources. I mean, for example, with Dalbergia cultrata, only less than 10% of the species range in being captured in this. And there's a real need to understand in the context of restoration these linkages between ecological and social contexts in the face of climate change, and the work that's been done in the project to support that will be hugely valuable in the next stages of going forward in improving decision making on restoration. Coupling that with the really important genomic work that Henry presented there emphasizes that we really have to be able to, you know, make decisions based on the adaptive variation of these species, and not only showing how that links to climate change in terms of conservation, but also in improving the outcomes of restoration through better matching of adaptive variation to sites, and particularly things like predictive provenancing that can really make sure that restoration interventions are resilient to climate change. So I think that part of the project alone has got huge lessons that can be transferred both to other countries in the region and also to linking these kind of the work on decision making to existing platforms for decision making that link species distribution with genomic data will be really pushing forward the boundaries in that kind of work on restoration decision making. Moving forward to filling the gaps on conservation, the project's really, you know, broken new ground in terms of the scale of identifying conservation areas in natural forest, which has been hugely important, as shown by Shalon and Bansa in Laos, and also by Dr. Tra Tri Hoa in Vietnam in identifying these natural forest populations that are really important for conserving genetic resource base. But emphasizing that a message that comes from this is that many of these sites don't retain a lot of mature trees anymore. So there's the importance of that moving into the future and making sure that these trees persist in the future. And reflecting back on the quiz from the morning of the huge value of the rosewood in terms of global trade, and yet the rather low value of the resource in terms of the genetic resources of the seed collections, that still remains a real challenge in being able to develop sustainable value chains for the genetic resource base, despite the high value globally of these species in the region. And I think critical to that has been the work that's been pushing forward on the capacity development and the training in not only seed collection, where we've seen in Laos over 5,000 seedlings being moved as part of the training in seed collection and transferring seedlings to community nurseries, but also over 7 trainings in 3 years in Vietnam, and not only the training in the seed collection and the nursery practices and the propagation techniques, as nicely illustrated by Banza, but also in thinking about the innovations in the entrepreneurship of seedling propagation and income generation, so nicely demonstrated by the presentation of Sote for Cambodia. And in this context of a hugely growing area of community-based forest management, 500,000 hectares now being designated in Cambodia alone, and the role that farmers can play in scaling seed collection, production, and seedling propagation as part of the sort of national initiatives in increasing the value of Dalbergia. I think it was really nice to see that this issue of increasing on-farm seed mother trees as well, because we recognise this is going to be a big bottleneck in the capacity to generate seedlings at scale. And the project has provided amazing continuation on past work to improve both the capacity and the knowledge on how to propagate climate smart or climate resilient material for restoration. It's clear that there still needs to be a lot of work done on increasing the demand. We see continued demand for Dalbergia timber, but you know, the questions remain about how we can increase the incentives for people to do more seed collection, more propagation on farm. And I think the work in this project really provides solid foundations on how restoration commitments at national and regional levels can be leveraged to increase livelihood outcomes for forest communities and diversified livelihoods from more sustainable use of forest genetic resources. And if you can't do that with Dalbergia, then it's something that's going to be difficult to do with anything, given the huge value of this group of species. So I see the lessons from this project being hugely valuable for not only Dalbergia in Asia, but other countries, but also more broadly in terms of the incentives to value and use tree biodiversity for sustainable development. And I think there were some comments made earlier as well, not just about this stage of the seed collection, propagation and nursery, where we've seen great improvements in the capacity and nursery infrastructure in these countries as well, but especially to ensure the long-term persistence of these trees in the landscape. So I think that's going to be our next stage of really bringing this work together to link those incentives for propagation and cultivation to more improved conservation of the trees and persistence of those trees in the landscape. So I think I'll stop there. I hope that's been useful and look forward to hearing further discussion.
Thanks very much, Chris. I'd like to reiterate what you said at the start, in the sense of I'm amazed how much has been achieved through this period of COVID. And I think it shows a great commitment from all the project partners in terms of that to achieve so much. And I'd like to congratulate them all on the efforts. I think also it shows the benefits of working in a number of countries. Species, as we know, don't respect country boundaries, but it's been very interesting to see across the distribution of Southeast Asia that the differences between species, or sorry, differences between countries related to how humans interact with those species. And so we've seen different examples from the different countries because of the different political systems, the social context and everything. And I think that brings quite a few lessons to learn. I think there are challenges that we have to face in terms of how we go forward, and how do we make sure that the lessons that we've learned from this project are still implemented, the conservation goes on, and that's a challenge to all of us to keep these things going, which depends on the collaborations continuing and continuing to look for funding for these efforts. I'm not sure if anybody else wants to make a comment. We're running out of time, but perhaps if anybody would like to make a comment with respect to those comments, it would be time now. And if not, I will hand you over to, I think Char is wanting to, we have the next sort of, before finishing with Rena, or maybe Rena's going to introduce the poll. Yes, the closing and everything. So thanks from me to everybody for their contributions, for participating, and I'll hand you over to Rena.
Okay, thank you very much, David, and also Kim Sobon and Chris for those insightful comments. It was really nice to hear how the results of the project link to both what's happening in the countries and also to the higher level, to the international negotiations and discussions about climate change and tree planting and so on. So we are now coming to the end of the workshop, and I would like to really warmly thank all the presenters and also all of you as participants for these vibrant discussions that we have had during the day. I hope that you've enjoyed the presentations and the discussions as much as I did. Personally, what I also really enjoyed seeing was all these photos about those beautiful lush rosewood forests across the region, and all those new seedlings growing in the nurseries. So this was really kind of medicine for the eyes during these times that we have been spending mostly behind closed doors and on screen during this COVID pandemic. So this demonstrates also what we would stand to lose if these species actually disappear further from the landscapes. I wanted to highlight in this closing few things that I thought were really interesting in the discussion and that kind of link to the conversations that we have had during the project. One of those was the role of local communities. And I realize now that there is no need to further underline that. It has been actually coming up in many of the presentations and in many of the talks. So the need to collaborate with local communities in terms of conservation, since they are the custodians of the land, they are the holders of knowledge about these species, their income and livelihood depends on the land. So we'll have to find ways actually to support what they are doing instead of setting up new protected areas on their lands. The other thing that I wanted to highlight was the value of regional collaboration, and David has also already alluded to that. But I think that it was really valuable in the sense that the species are so threatened, and that the kind of the need to improve the conservation status and to restore these species is so urgent that through this regional collaboration and through the exchange of experiences and lessons learned, we have actually been able to kind of accelerate learning about what works where in terms of conservation and restoration of these species. So I think that this is something that has been really valuable, and I hope and I believe that this continues beyond the project. But it's also something that has made this project so much more pleasant to implement and to collaborate on. So we now have a network of rosewood experts across the region, and also a network of friends and colleagues to rely on as we continue these efforts for the conservation and restoration of these species. So yes, would be very nice to hear from all the rest of you, and continue these exchanges that we have started during this workshop. So it will definitely take all efforts to help protect these species. So I think that with that we can formally close this workshop, but we wanted to keep a little bit space at the end for actually additional networking if there were questions that you wanted to ask or comments that you wanted to make, but you did not have a chance for before. So we close the program formally now, but the presenters, the speakers will stay here for another 10 minutes. So feel free to reach out to them. You can raise your hand and speak up, or you can write comments in the chat, either directly to the presenters or to everyone in the meeting. So with that, thank you again for joining us today, and yes, have a pleasant evening.
Rina, would you want to take a picture of everyone? We can like open our cameras and we can take a picture.
That's a wonderful idea. Let's do that. Can I ask everyone to please open your cameras? Char, I think we can count on you to take the screenshot.
We have 2 frames, so I'm gonna count. 1, 2, 3. Another one. 1, 2, 3. Thank you, everyone.
Thank
you.
Thank you. Thank you very much. Have a nice day.
Thank you. And the results of the project are also available on the website of the Asia Pacific Forest Genetic Resources Program. So those can be visited there. Char, maybe we can put up the video about the publications and then followed by the other videos in case there are no questions. Thank you.
Thank you.
I say in the chat, there's one question that I think has not been answered. So Mr. Wutha from Cambodia was asking whether there are any techniques of genetic processing or improvement to make Dalbergia trees to have less branches. So if any of the project participants or partners would like to take this, there is still a bit of time. Thank you.
Rina, actually, I answer that question. I suggested to collect seed from the good seed source in Siem Reap province because based on our experience, we collect seed from the plantation and we plant it in the southern part of Cambodia. The tree does not produce branch, produce less branch compared to other source. And I also suggest to apply silvicultural treatment in the early stage of the planting. I mean that maybe start from year 1 or year 2. Well, start pruning. Thank you.
Okay, thank you, and apologies, I had missed that. Yes, so hopefully that answers the question. Okay, if there are no further questions, we will close the meeting now. So thank you again, and I hope you enjoyed the event. Please keep in touch. Thank you.
Thank you. Thanks.
Thank you. Hey, everyone.
Thanks, everyone.
Bye-bye.
Bye.
Bye.
Bye bye. Thank you. Thank you.
Thank you. Thank you.
Thank you.